Thank you very much, Jeff. All right, moving on to our next presentation, we have Dr. Mark Avigan. Dr. Avigan comes to us as the Associate Director in the Office of Pharmacovigilance and Epidemiology at the FDA. He is a clinical hepatologist with expertise in drug safety science and cellular regulation.
Well, thank you, Nick. I'm going to take a step back and talk about the meat of the DILI challenge in drug development and get a little bit under the hood about the complexities, mechanisms, and then move to where CIVMs, if we can develop them, will make a difference. So this is a broader talk. And again, this is going to be, if we're going to be successful, this will be a multidisciplinary effort across disciplines. So we have to remind ourselves that DILI has been a major reason of hepatotoxicity for removal of many drugs, over 50 drugs. And although in recent years, and this is a list on the left side of some of those drugs removed from marketing, in recent years there haven't been that many drugs removed, but DILI and hepatotoxicity continues to be a main reason for disruption in drug development. And there is an imperative to get better at early prediction. And in conjunction with that, and I apologize for the point that many drugs now, emerging drugs, continue to have warnings and have constraints in their marketing across different kinds of therapeutic groups. And as we discuss, I see we're a little cut off here, but but the main point in this slide, hopefully you'll see, as we discuss DILI, we have to be really mindful of the emergence of new categories of products over time. We have to know what we're dealing with moving forward. And there are many studies on this, but the key point is that there's an emergence in recent years of many more cancer drugs and biological agents in the mix of approved products. So we have to be able to consider DILI mechanisms in those products as well. And this is a study from a listing of the approved products in 2021, a recent listing, and you can see that many products now are in the biological category approval, monoclonals and enzymes, recombinant enzymes, and many kinase inhibitors that target specific cell surface receptors or intracellular phosphorylation enzymes for metabolism and so on. And so when we think about hepatotoxicity, we have to think about where those mechanisms associated with these kinds of products may need to be interrogated. And so irrespective of the product type, when we consider a drug that may have a hepatotoxic profile, there really are four domains that we think about. The mechanisms, and we'll talk about those in a moment, but we're talking about different kinds of mechanisms, the PK/PD profile of the drug, and whether there may be excessive levels above a threshold of toxicity in liver, the adaptation effect that that people have, the livers have, to make a comeback, to cytoprotect, to regenerate, and where there may be failures in some individuals, and then susceptibility factors in individual patients, both genetic and non-genetic. And each of these are areas of large investigation now. So this is a very large field in the literature. So a key point in this talk is that different kinds of, there are many different kinds of DILI that actually, mechanisms that can explain DILI with different kinds of products. These include the accumulation of reactive metabolites that induce oxidative stress or mitochondrial injuries or different kinds of inhibitions of apical transporters, BSEP as an example. Other examples with idiosyncratic injuries are the induction of adaptive immune responses to a response to HLA-presented metabolite, drug metabolite on the cell surface of a hepatocyte, for example, in the presence of a dangerous signal, which is in the background. And then in immunotherapies, which we've seen more of, there's a loss of that tight homeostatic balance between autoreactive cells and Treg cells, the cells that keep them in check, normally keep from being overly active in the normal state. And there are other mechanisms as well. There's another slide I was going to show you, but for sake of time, we won't go there. But it's not a surprise, therefore, that we're dealing with a variety of phenotypes, clinical phenotypes in the clinic when we see patients with DILI. Acute liver injuries with hepatocellular forms of injury and various kinds of clinical phenotypes. We see cholestatic events that are either acute events or more chronic events, and then we see persistent events. So we see a whole smattering of different kinds, not only of mechanisms, but of clinical pathologic phenotypes. And these have been conveniently categorized for us in recent times by, this is from a paper by experts Bjornson and Hufnagel, that have put them into buckets of direct injuries. These are, an example would be acetaminophen. These are dose-related. These are predictable. Indirect injuries, which are effects of the biological characteristics of the treatment, but they're unintended effects, and there's a whole variety of those kinds of effects. And then the idiosyncratic forms that were mentioned by Jeff, where these are sort of acute thunderstorms in patients that usually they're not predictable, and they're usually related to an HLA association that has to do with an adaptive immune reaction. So again, from the clinical point of view, what we're interested in public health are the life-threatening DILI outcomes, which are acute liver failure, but also other kinds of outcomes, vanishing bile duct syndrome. There are chronic liver injuries with hepatic dysfunction and decompensations in patients who already have underlying cirrhosis. And in clinical trials, which is the best way to see an early DILI signal in terms of a prospective controlled manner, there are two kinds of signals that we see. We see imbalances of ALT effects or liver test effects between study drug and comparators, and then we also see more significant injuries where there may be jaundice, there may be liver abnormalities that require case-level causality assessment to actually determine whether the drug actually is causing the event. And it's in this category that we have you may have heard the term Hy's Law. These are hepatocellular injuries with jaundice, that are caused by a drug, which are harbingers of a potential serious problem in other patients. And there's a DILI guidance that's been out there for a number of years. Some of you may have come across it, which underlines the predictive value that there's a Hy's Law case, a hepatocellular injury case, in the mix of clinical trial patients. That's a bad sign. If that liver injury was caused not by the drug and not by alternate causes, which are systematically excluded. And I'm going to just go, I want to get to the CIVM part of my talk, but I want to give you an example. This is a drug that zimilgatran was a thrombin 2 inhibitor that was being developed, and it essentially had a idiosyncratic DILI profile associated with it, associated with DRB1, 07, HLA2. Patients were positive for that HLA. And you can see that it was associated, just on the listing of the points about this slide, with a huge signal for liver injury with advanced injury in the clinical trial, called SPORT-T5. And it had also a very robust elevation of ALT. 8% of patients had elevations of ALT. So this is the kind of thing we were trying to early on determine in clinical trials, and and the question, of course, is can we do a better job early on in the clinical trial progression? We'll talk in a moment about that, identifying drugs and negating them out early in their development. And this is what you see in the clinical trial phase. This is what's called an E-dish plus 2D graph, which bilirubin ALT, and these are the peak levels during treatment. You can see in that right upper quadrant, which are abnormalities of both ALT and bilirubin above a certain threshold. And you can see most of those are the triangles, which are the active treatment, and just versus the comparator group. And this is the kind of case that you want to absolutely avoid in a clinical trial. This is a 80-year-old gentleman in the trial who on day 56 started inkling up their ALT levels on this treatment, and then very rapidly developed almost like a storm where there was an adaptive immune reaction and elevations of bilirubin, developed submassive necrosis, and even though the drug had already been stopped as per protocol, the patient went on to get liver failure and died. And this is the kind of, again, the clinical profile that we see in terms of the signals that we see in clinical trials at the clinical trial phase, where there's an elevation of bilirubin. That's a bad sign. This is what Hy Zimmerman pointed out in his early research on this, and this is borne out to be true. Now, I want to just make a couple of points. I mentioned there are other clinical pathologic phenotypes. We're not always talking about idiosyncratic reactions. There are also cases of hypersensitivity, for example, with the anti-epileptic aromatic drugs, or with, let's say, sulfa drugs. Those are different phenotypes. These are very rapid onset reactions. There is memory with re-exposure, so there's an amnestic shortening exposure response. There's immune-mediated hepatitis, which is a different phenomenon where, let's say with checkpoint inhibitors, where the patients actually have loss of homeostasis and they between the various networks of T cells, and then they get an autoreactive reaction, so that has a different temporal characteristic. And then there are reactions which don't injure the hepatocytes, but rather injure the bile ducts, as in vanishing bile duct syndrome. And just, I'm going to finish on these clinical phenotypes as examples, just to show you that we're not just talking about one kind of injury. So this is an example. This is from the NIH DILI network. When they went back and looked at their first 1,000 patients who were registered as DILI patients, you can see that there's a certain percentage of patients had these duct injuries. These are not hepatocellular injuries. It's a different target cell. And some of those are more pronounced than others, but it's important to know that. And we have recently seen some examples where that's the most pronounced injury in the development program, so the injury of the most concern. And this is a drug, it's a CSF1 receptor inhibitor called pexidartinib to treat TGCT, which is a non-malignant tumor, that where CSF1 is duplicated and the levels are very high, and so these patients with the treatment of inhibition of their receptor, the CSF1 receptor, there's a very pronounced rate of liver injury, and the patients who've run into trouble are these patients on that bottom row, where you can see that they develop a kind of mixed or cholestatic injury, and about 5% of those patients in clinical trials, and some of them have had what's called vanishing bile duct syndrome. They lose their ducts and the bile ducts, and they don't come back. And this is an example of what a patient who had that effect from a exploratory study, and this was a patient in a breast cancer trial, and she ended up with a liver transplant after 20 months, just smoldering progressive disease. So this is what we're concerned about. So now let me now circle back to what we really want to measure early in drug development if we can. And there really are some key measures. We want to understand the drug liability, that's the predicted hazard, the drug-associated risk for serious liver injury, the utility of liver test monitoring in clinical studies, and then if we can, think down the line, what would be the real-world DILI burden in the post-market, if we could have accurate measures for that. And this is an iconic kind of figure taken from a nice review by Richard Weaver and Jean-Pierre Valentine, who are toxicologists in drug development. And it makes some key points. And again, that top vector is, you've all seen it 100 times, but the key point is in the development program, it's like a football game. You want to quickly whittle down many candidate drugs to one or two optimal drugs for candidate, and then bring them into the clinic. And along the way, you want to be able to, as early as possible, identify safety liabilities to gate them out. And then in clinical trials, if you see a signal, a liver test signal, you want to know, is that of significance, or is that just kind of a little smoke but no fire? And the bottom row here shows that the three big safety areas in drug development that are disruptors, that cause disruptions, that cause discontinuations of drugs, are the cardiovascular, the nervous system, and the hepatic system. And we're talking now contemporary. And so you can see, and hepatotoxicity continues to be a problem at not only the early phases, but the later phases. So if we had tools in place that could allow us to sort of have a better sense of what we're dealing with, that would be terrific. So the goals in drug development would be, A, to efficiently weed out drugs with unfavorable hazard, okay, and using a variety of techniques to do that, or methods to do that, to reliably predict a candidate product's DILI risk profile, to clinical trials of a target population, and to strengthen the accuracy of DILI risk prediction in early clinical trials. Where patients are already underway, you might see a liver signal. You don't know whether it's going to be, again, just mild injuries that always get better, or whether there is going to be a potential for more serious injuries, which is what we talked about, the more serious outcomes. So, so this is where the question is there complementarity with the ability to look at, and my colleague, Dr. Skip Hayashi, tomorrow will talk a little bit about this in the clinical context of review of DILI, potential DILI. When there are questions like this, we often get a kind of portfolio of data around preclinical methods. This is commonly done with our colleagues in the NCTR, Dr. Minjun Chen, and these are some of the kinds of tests that the clinical reviewers will look at also to see, how convincing are they. In vitro toxicity in vivo animal toxicity testing, the reactive metabolite measures that some of you may know about, the mitochondrial functions and membrane transporter interrogations, the profiles of reactive species, and then the physiochemical properties of the drug. Is it lipophilic? What's the dose? And so on. And you can, from that, get a sense. Sometimes these are actually very predictive and valuable measures, but sometimes they fall short and actually give misleading results. So by themselves, they're, as a group, we look at these, but they're not always validated. And Dr. Hayashi will talk about this more tomorrow. So a number of years ago, I wrote a book chapter with a colleague of mine, and we just went through all these methods of potentially hepatotoxicity of the drug, and we just sort of what's out there, and we sort of lined them up. And they all, each test system, and I'm not going to go through these because there's a bunch of them, but they have utilities, but they also have limitations and gaps. And that's the main thing that so methods on the left in red, and then limitations gaps on the very far right. And you can see that often some mechanisms are represented by a particular test or method or approach, but other mechanisms for DILI are not in that frame. And that's a key point for this talk. And so these are the kinds of in silico or in vitro methods. We heard about single cell cultures from Jeff, that they're limited in their discrimination among DILI and non-DILI associated drugs. So with these limitations, we have now the evolution of these CIVM and other approaches. And we've heard what some of these are, and in the literature, and there are different groups working on these. There are hepatocyte-like epithelial cells that are derived from macrophages that are being developed by a group in Germany there. And this is still a work in progress, very interesting for a diagnostic tool from patients' samples directly. 3D organoids of the inducible pluripotent stem cells. It's a very exciting area where the stem cells are reprogrammed with the Yamanaka factors, and then you can interrogate them. They're grown as hepatocytes. We'll hear more about the microphysiologic, microperfusate systems of so-called liver in a chip. And then there's a whole new area of spatial transcriptomics, which is very exciting. And I'll show you in a moment, that actually interrogates the liver, the sections themselves. And then cellular and molecular biomarkers that measure the state of hepatic immunotolerance, when loss of immunotolerance is the reason for the injury. But even with these tools, there are going to be some blind spots that we have to be cognizant of as we develop these tools that you should think about. One is that idiosyncratic hepatotoxicity is often linked, as I said, to HLA. So the HLA has to be represented in the assay, because otherwise you're not actually looking at that question. Many patients, certain drugs disrupt immune cell networks that are normally homeostatic, and that actually causes the adaptive reaction. So again, that our testing right now doesn't necessarily look at that particular kind of step. And then for certain diseases, there's there may be changes, underlying liver diseases, NASH and hepatitis, that may change the ability of the liver to adapt when there's a perturbation, to cytoprotect, to regenerate. So those again have to be thought about. So I want to just finish and summarize the challenges. We already talked about the multiple mechanisms and clinical pathologic phenotypes. There are complex in vivo architectural determinants. There's a complexity to the architecture of the normal liver that includes multiple cell types, and also the idea that there are gradients of drug metabolism across the hepatic cord. That you have to think about, if you're a CIVM, does it actually represent a perturbation in that effect if there's a drug metabolism problem or clearance problem? And this is just a nice picture diagram from a group at Vanderbilt that sort of captures this complexity of multicellularity in that diagram at the bottom. There are 4 or 5 different cell types that are organized, and the gradient of hepatocytes as they march down very slowly from the zone 1 to the zone 3. And you can see it. What's the key thing in this diagram are that the different activities of enzymes that may have an effect, direct or indirect, on toxicity, including cytochromes not listed here, but they would be on the zone 3. If there's a perturbation because of an underlying effect, then that might be a trigger for why certain individuals would get DILI. So again, you have to remember what the potential complexities are in the model. And then I'm going to finish with 2 slides, but there are other considerations with a CIVM. First, you may need, in an analysis, in an assay, you need controls, and you need you may want to test multiple conditions or multiple drugs side by side. So it has to be practical and cost effective. These are really hard to set up because they're really complicated multicellular things that you have to think about the practicality and the cost. The second point is that the design of a CIVM-based assay should reflect the likely mechanism of toxicity that you're thinking about, and also and simulate the in vivo blood and liver drug concentrations. The third point is that if you're going to have an assay with a readout, let's say of cell death, the readout has to be on a scale or a range where the scale is broad, so that you can sort of see differences between samples. And when immune-mediated hepatotoxicity is an underlying DILI mechanism, then it might be useful to actually look at inflammatory cytokines that might emanate, say, from Kupffer cells or immunocytes that are actually important steps in the mix of toxicity. And then the very last slide, but this is again, again, these are just things, since this is a group of toxicologists who know bench research. But this is an important point: is that the confidence of results in results of a CIVM-based assay depend on reproducibility as well as positive and negative controls. So those controls will be different depending on how you frame the question. But for example, you might, if there's a known effect with a certain class of drugs on DILI toxicity with some members, but not with other members, you might want to line them up as controls. You might want to test the vulnerability of your assay system to hepatotoxicity with direct hepatotoxic drugs as another control, such as high dose acetaminophen versus therapeutic concentrations. If you're testing DILI susceptibility, you might want to think about HLA. And I think if we go forward, I think this problem of HLA matching will eventually be overcome. I think but it's something that has to right now be thought about. And then the last point is that for some DILI effects, there could be demographic as well as drug disease and drug-drug interactions that affect susceptibility. This is an area that we're very concerned about with our DHN colleagues who are in the lead on this, looking at patients with NASH, for example, who might have susceptibility to certain drugs, as well as chronic hepatitis. And with that, I want to thank you for your attention.